Okay, everybody, here we go. This lesson 2.1 has to do with DNA. And what you're looking at here is a very famous X-ray of DNA. And this X-ray allowed the folks who figured out what DNA was made of and what it looked like a really good opportunity to figure out its shape. And you can see that X down the middle, and we'll allude to that later. But anyway, this was a very important picture. So as far as this lesson goes, all right, all living things have the ability to pass on genes, or genes in DNA, which influence the traits of future generations. All right, so the essential questions: what are the contributions of various scientists to the discovery of DNA molecule and its structure? And there's some examples. And then how is the information stored in DNA? And how do we use it? All right, so the objectives describe the contributions of the various scientists to the discovery of DNA molecule and its structure. Hershey and Chase, Watson and Crick, Chargaff, Franklin, Wilkins, the Human Genome Project. Explain the scale and relationship between nucleotides, genes, chromosomes, and the genome. And describe how DNA can be extracted from a cheek cell and how it can be used by a scientist. So if we go to the presentation, we'll just kind of go through it. All right, so the role of genetic material. Genetic material must carry out two jobs: duplicate itself and control the development of the rest of the cell in a specific way. And Francis Crick, they're a very famous scientist, and talk more about him in a second. Any of these links that are in blue usually have a video associated with them too. So you might want to go through the presentation and click on them. All right, the information necessary to sustain and perpetuate life is found within a molecule. This genetic material that is passed from one generation to the next is a blueprint for the building of a creature, a living organism. If you remember in Jurassic Park, that's what they needed. In the 1850s, it was unthinkable for the leading scientists and philosophers that a chemical molecule could hold enough information to build a human. They believed that plants and animals have been specifically designed by a creator. So early scientists, Charles Darwin, 1859, he was the evolution guy, challenged the idea that a higher being created life, and life changes over time through natural selection. And so there's a constant change to it. The Mendel in early to mid 1800s discovered that how pea plants are able to pass on their genetic information, right? And traits were passed on through factors. We call them today genes. The question became, what are these factors? At that point, we weren't able to look inside of cells and just, what DNA wasn't even discovered. And then we have later on the 1800s, Friedrich Miescher, a German scientist, and he isolated the chemicals found in the nucleus. These were proteins and nucleic acids. And so, again, you can click on the link there to find out more about that. Okay, some other people that were involved in the history of DNA. All right, we have Oswald Avery, Alfred Hershey and Martha Chase, Chargaff, Wilkins, Franklin, Pauling, and Watson and Crick. All right, so we'll just kind of go through some of these individuals. All right, known as the transforming principle, Frederick Griffith and Oswald Avery, using bacteria, showed a lethal trait is passed on from one organism to another. DNA was the molecule of inheritance, but unsure how it worked. So always a question about whether it was a protein inside the cell or the nucleic acid DNA. That was the inheriting factor. So Alfred Hershey and Martha Chase, 1952, they used a bacteriophage, and you can see the picture down there. It looks like a rocket ship, but basically that is a virus. And the DNA that you see in blue is being injected into the cell. All right, so they were able to recognize and prove that DNA was the hereditary material. The bacteriophage was the ideal organism for settling the debate between DNA and proteins, which one was going to be the factor that we passed on after generation after generation our traits. All right, the bacteriophage is a virus that infects bacteria. The DNA is inside that protein coat. And then the question question, what infects the bacterium, the protein, or the DNA? So that was the question they were asking themselves. So if you look at this picture, all right, on the left-hand side, you have the virus and the sulfur labeled protein capsules. So they put a little identifier on the protein outside of the virus, and then they had the virus infect a cell. You can see the DNA was shot into this bacteria right here, all right? And what they recognized, since the protein was not being injected inside of the cell, it was the DNA, there was no sulfur found in here. They did the same thing over here, and they infected the DNA with some phosphorus, and they recognized, once the DNA was inherited inside of the bacteria, that they were able to find phosphorus inside the cell. So that is how they discovered it was not protein, but it was DNA that was going to be the inheritable factor. So here's a very important scientist. Her name is Rosalind Franklin. Back then, she was a female in the science industry and not given a whole lot of respect. This is over in England. And like her laboratory would be in the basement, and she just wouldn't get this funding that the men would get and all that. But she wound up doing great work. And she had the ability to take X-rays and take a picture of the DNA. And this photograph, like we said earlier, helped determine the structure of DNA. What did it look like? And she was working with X-rays. Back then, they didn't realize X-rays potentially can cause cancer, and she died very young of cancer. She didn't take precautions like you see X-ray technologists do today with the lead coating or stepping outside and where they take a picture and all that stuff. All right, trained as a chemist, was expert in deducing the structure of molecules by firing X-rays through them. Her images of DNA disclosed without her knowledge, put Watson and Crick on the track towards the right structure. So Watson and Crick were able to get that photograph from an associate who was working with Rosalind Franklin named Wilkins. And she did not want that to happen, but he was able to grab her photograph and give it to them, and that helped them finally recognize the structure of DNA. Okay, so Erwin Chargaff, what role did he play? He figured out, all right, he discovering that these pair of nitrogenous bases, A, T, C, and G, right? A and T were always found in equal quantities. C and G were always found in equal quantities. A stands for adenine, T stands for thymine, C stands for cytosine, and the G stands for guanine. The equal amounts was very critical. So if you had 100% of something and you knew that 25% of it was A, then 25 had to be T. It also told you that 25% of that potion or stuff was going to be cytosine and 25% would be guanine. Okay, so knowing that they were found in equal quantity was very important to Watson and Crick in the discovery of DNA. And then finally, we have Linus Pauling, 20th century chemist, led the way in working out the structure of big biological molecules. He was a competitor to Watson and Crick, and he initially thought that DNA was a triple helix rather than what we'll see here that Watson and Crick figured out. And that really worried Watson and Crick because it was a race to figure out the structure. All right, so now these two gentlemen, an American and a British physicist, working together over in England. All right, Watson presence at the conference in Naples in 1851 saw the X-ray by Franklin. This made a strong impression on Watson. Genes might have a regular pattern. 1952, Chargaff visited Watson and Crick and explained how the base pairs A and T and C, G were complementary, always found together. Crick convinced the pairing of A and T and C and G is the basis for coding genes. What they deduced from the X-ray was that it was a double helix, not a triple, always the same width, 2 nanometers, and the bases were stacked 0.34 nanometers apart. Chargaff's rules: adenine always pairs with thymine and cytosine pairs with guanine. Watson and Crick published their findings in Nature. The paper ends with, it has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. And that was a picture of their model. And that's Watson and Crick there. You can see the DNA structure in the background, or what they're pointing to. So James Watson, Francis Crick, Maurice Wilkins received the Nobel Prize in 1962 for medicine. Rosalind Franklin, a colleague of Wilkins, and produced the image of DNA that helped Watson and Crick with the structure of DNA, died in 1958 and was not initially recognized with a prize, but people found out later on who she was and what she did. Okay, and that will be the end of this very interesting lesson and an important one.